Vande Matram children, welcome back to my YouTube channel RSK Competitive World. So today we'll discuss another new point that I will come back to afterwards that. But now just I want to revise in which the previous sessions we discussed some points regarding heterotrophic mode of nutrition. Okay, we discussed in details that is autotrophic mode of nutrition. That is only one thing, that is the plant is autotroph, means it will prepare, it's the substance which will prepare its own food, we can call it as a autotrophs, and this process is called autotrophism. So not all plants, most of the plants. Okay, and also parallelly to this one we discussed heterotrophic mode of nutrition. In that again we divided that heterotrophic mode of nutrition, which are those parasitic mode of nutrition, saprophytic, okay, then symbiotic, then insectivorous. So these are some other kinds or types will comes in a heterotrophic mode of nutrition. So we discussed these in detail in the last session. No need to discuss, just I want to revise it, that's it. Okay. Very good example for the parasitic. You know that these parasitic, symbiotic and saprophytic and insectivorous, these kinds of plants will not prepare their own food, will be get their food by other means, by other organisms, will depending on other organisms. That's why we call them as a heterotrophic mode of nutrition. Okay, yes, we'll move to the next slide, that is today's session. What we need to discuss here, how nutrients are replenished in this soil. I want to discuss that. I want to give some meaning about this replenished. The meaning of replenished is nothing but to rich, to make the soil rich by adding nutrients. Not only by adding nutrients the soil will not become rich. Again we need some gap between two consequences of crops. Are you getting my point? Continuously we can't get a better yield of a crop if you keep on getting, that is if you keep on start sowing the seed into the field, you can't get a better yield of a crop. We need to give some that is a gap between the crops. So then only we'll get, we need to add manure, fertilizers, and that also again we need to leave at least 3 to 4 months completely empty the whole land, the field. That soil must need water and much of sunlight also. Then only it will become, the soil become a porous. And easily after becoming porous, by adding manure, it will become porous and will exchange the gases, which some microbes will be present in the soil. Those microbes are nothing but a small living organisms also will respire, means respiration will take place. They will also exchanging the gases. They will also need food, no, to survive. So for that soil must be rich by adding nutrients, manure. So all nutrients will be getting it by adding manure and fertilizers. That I will discuss. Okay, see, plants absorb nutrients from the soil, so the nutrients in the soil decreases. So you know the plants must need nutrients to better growth of its whole body. So if it start absorbing the nutrients which are present in the soil, then there will be automatically there will be a decrement in the nutrients in the soil. Okay, so that's why we need to keep on adding, that is manures and fertilizers to the soil. Are you getting my point? Are you understanding everything? So the farmers, what he will do is that whenever there will be a decreases the nutrients in the present in the soil, that afterwards what he will do, he will keep on adding manure and fertilizers. Are you understanding the meaning of manure here? Manure is nothing but it is also fertilizer. But there is a little bit difference between this fertilizer and manure. The manure, it is a organic matter, that is does not contain any chemicals. Means, good example, the manure is made by animal dung. Animal dung. It's a natural one. Okay, but fertilizer, it is made up of chemicals. Chemicals. Are you getting my point? This fertilizer completely depending on, that is, chemicals. These are made up of chemicals. So many chemicals which are prepared in laboratories. And there is lots of difference between the manures and fertilizer which you learn in the upper classes. I will come back to you afterwards. Got my point? The farmers will keep on adding the manure and fertilizer which he want to increase the fertility of the soil. That is nothing but a replenishment. Okay. See, the next para, the bacterium called rhizobium, which lives in the roots of leguminous plants like gram, peas, beans, etc., convert nitrogen from the air into the soluble form in the soil and makes the soil rich in nitrogen. So please keep in mind that I explained this in the previous session only. I explained about this one, how replenishment of a soil will become. Okay. So that some of the leguminous plants are there. They have given some examples here: grams, green gram, yellow gram, black grams. So many grams are there. Peas you can get. Okay, green peas, beans, so many kinds of beans are there. So these are why the rhizobium bacteria are present in the roots of these leguminous plants, because these leguminous plants contain much of nutrients. Their roots contain much of nutrients. Are you getting my point? So these rhizobium bacteria will convert that nitrates into soluble form, means which the plant need. In that form it will convert and it will absorb it through the xylem vessels. Are you getting my point? Which afterwards it will transport that nutrient to all parts of the plant. So I explained also, this is a symbiotic relationship between that rhizobium bacteria and the plant. That plant gave shelter and food to the bacteria. In return, bacteria is converting its food, that is in the form of nitrogen, that is nitrogen to nitrates, into soluble form. Are you getting my point? This is symbiotic relationship. That what he explained here. In return, the plant provides food and shelter to the bacteria, so they have a symbiotic relationship. Plant will give shelter. Got my point? It is shelter, nothing but it is a home for that. Are you getting my point? It is a home, and it will give food also for that bacteria, and bacteria in return, it will also help the plant in the form of transporting nitrogen, that is nitrates, nothing but nutrients to the plant. Okay, in this way the soil can be, we also by adding manure and fertilizers we can replenish, or we can make it as a rich in nutrients. I hope you are understanding everything. Okay, we'll move to the next slide. The next slide, that in your lesson, the nutrition in plants, we have totally activity 1.1 and activity 1.2. That last session, this is the last session, that we are at the end of the chapter, and we will discuss in detail this activities, very important at the point of examination also. See, that activity 1.1, according to the NCERT syllabus, that is to show that sunlight is necessary for photosynthesis. Will you agree that sunlight is necessary for the photosynthesis process? The first session only I discussed it. Sunlight is very much necessary for the photosynthesis process, that you know very well. What is photosynthesis process? My question is, what is photosynthesis process? The plants like autotrophs, they will prepare their own food by absorbing sunlight, carbon dioxide and water. Okay, in presence of sunlight, the plant will absorb sunlight, carbon dioxide and water, and it can prepare its own food in the form of glucose. At the same time, it releases the oxygen. That equation also we saw it in the previous session. So this is a photosynthesis process. So to complete this photosynthesis process, that is a preparation of food by a plant, sunlight is very much important. So So that what we have to discuss here, how it is important sunlight, is it important or not, that what we have to discuss here. So finally, after completing this activity, we can conclude whether it is necessary for photosynthesis or not. Okay, yes, see, take two potted plants that parallelly you can do in your home. After reading this one, after watching this video also, you can do it in your house, all of you in a home. And it is a better best time now you are getting, you can do this activity in home. In upcoming days, I will show this activity also. We can do this practically also, we can do it. Okay, yes, take two potted plants of the same kind, keep one in sunlight and other in the dark room for 2 to 3 days. Then take a leaf from both the plants and test for the starch. Why we need to here test for starch? This is very important. Why we need to test for starch? Where should be the starch in the plant? Is that starch is there in the plant already? Yes, 100%. That we discussed the equation. What is that equation? See this equation. I will CO2 plus H2O. Okay, it will give C6H6 H12O6. Nothing but it is a starch. At the same time, it releases oxygen. Are you getting my point? See, this C is nothing but a starch. So the plant, that is whenever discussing initially in the first session we discussed, that is the lower surface, that is the lower epidermis. The photosynthesis process will take place in the lower epidermis, that is the chlorophyll, where it is present in the chloroplast. Where the chloroplast in the, where it will be being like a structures. What do you call them? Just recall it, please recall it again. You can see my videos, those. If you can't getting this one, again you could go for the first video. I can just you see, watch it. Okay, keep on watching it again and again, so which will become perfect. You just link all these slides, means all those videos you can link it again and again. You can watch it, see it, then only you can understand the whole lesson. Okay, yes, the starch is present at the lower surface of the, that is lower epidermis of the leaf. Okay, so we have to start, that is we need to take two potted plants and keep one in sunlight, another one should be in the dark room for 2 to 3 days. Then take a leaf from both the plants and test for the starch. So what we have to do is the next step, that is we need to take two potted plants and one we have to keep in the sunlight, another one we have to keep in the dark room. Then after 3 days, that to take two leaves from the respective pots, and what we need to do is we need to test for the starch. In which leaf the starch is there, meaning is that plant that absorbed sunlight, then only it can convert that into the starch, that is nothing but a glucose. Are you getting my point? So for this test we need iodine solution. That is, I hope that all of you 7th standard students, whenever they will be in the 6th standard, you did this experiment in the chemistry laboratories, that by using iodine solution, that is you have taken a small test tube, in that you used, I hope that you used bread in that, otherwise rice in that. You used the rice or bread in that, you inserted small pieces, you crushed it and you inserted into the test tube. Then afterwards, then you have added iodine solution to directly that. After, by adding small amount of water to that crushed bread, little bit, that is after by adding small 2 to 3 drops of iodine solution, that iodine, that is by adding iodine solution, it will be converted to, that is blue black solution. So that blue black solution, nothing but it indicates that presence of starch. Are you getting my point? So meaning is nothing but a rice, or whether it will be a bread, it contains the starch. Are you getting my point? Nothing but a sugar, nothing but glucose. So here also we are testing for the starch which is present in the which leaf, otherwise whether it will be in the pot which is present in the dark room or it is in the sunlight. Okay, that what we need to discuss, confirm. So put the leaves in the test tube. We have to take a separate 2 test tube for that, and one respect to both the leaves we have to insert in those test tubes. Pour some spirit in it. That here why we need to add the spirit here, because spirit is nothing but alcohol. We need to little bit we have to add alcohol. Spirit is nothing but alcohol, that is ethyl alcohol. We will especially will use for this one as a ethyl alcohol, and we can after adding, that is pour some spirit in it or alcohol in it and put the test tube in a beaker containing a water. That is, you know very well in the chemistry lab we have so many beakers that we have to take a beaker and you can keep those test tubes under the beaker, in the beaker containing water. Are you getting my point? Boil the water till the green color in the leaves comes out into the spirit. So after what? After that beaker containing a water which we inserted 2 test tubes which containing 2 separate leaves, which is one is in the dark room, another one is in the sunlight, and we have to start, that is we have to heat it. Are you getting my point? We have to heat it up to 5 to 10 minutes, which the spirit will completely removes a green color. The green color which is present in the leaves, that will completely removes. Then after what we have to do? We have to take those leaves with the help of pair of tongs. We have to take that separate 2 leaves and we have to wash those 2 leaves with the help of a tap water. Then we can place in the dish, separate 2 dish. Then what we have to do now? Then that leaves completely removed by its green color, converted to a white color. Now we have to take a iodine solution. Then 2 to 3 drops of iodine solution just drop on the respective leaf. Then the leaf of the plant kept in the sunlight turn blue black. Are you getting my point? After dropping 2 to 3 drops of iodine solution on the respective 2 leaf, the leaf which kept under the sunlight will turn to blue black. The leaf of the plant kept in the dark does not turn blue black. Before only I explained you, which you did in the 6th standard. That is, you have taken test tube, you crushed bread or rice with the help of adding the water. Then afterwards you can added some 2 to 3 drops of iodine solution, converted to a blue, that is blue black, little bit of purple color. Okay, purple color means the leaf which can be kept under a sunlight will turn to the blue black, that is after adding iodine solution. But the leaf which can kept under the dark situation, dark condition, that will not turn blue black. Means this experiment means by this activity shows that sunlight is most important for the photosynthesis process. Are you getting my point? Means if turned, the leaf which can be by adding iodine solution on that, it will turn blue black, nothing but it indicates that the starch will be present in that. The starch is there in that. Then only it will turn. That is iodine test. Okay, are you understanding my point? That this activity we can discuss in detail in the laboratories. I will show you also. Okay. So I hope that you understood this one. If any difficulties, so you can write that question. You can drop me to my personal number. Okay. Yes. We'll go for the second activity 1.2, to observe the fungus bread mold in the bread. That is, you have to take a small piece of a bread, then you have to moisten it with water. That is a small amount of water you have to sprinkle on the bread mold, that is piece of a bread. Leave it in a warm place for 2 to 3 days till fluffy patches appear on them. That is a fluffy patches. Here the word that is fluffy, nothing but it is a wooden like structure. It is. That is, in the form of wooden, that is wooden like structure, nothing but a small kinds of a wooden, that is nothing but wooden like structure means small like a wool like structure, nothing but it is a fluffy, nothing but it is a wool like structure, not woolen, it is wooden, it is wool like structure. Okay, so here patches, that is a wool, you some of you observed this one. How you observed? See this, whenever there will be a little bit in a rainy season, that you kept there your chappals for you will not touch your chappals at least to 2 to 3 months, I hope. Okay, so after completion of the your rainy season, you can again you will wear the some chappals that which are made of by the leather chappals. So whenever you keep on, that is you will go and touch that chappals, the time you can see that a small woolen like a structure, that is a thread like a structure. So that is nothing but a, we can say that is a fungus. Are you getting my point? See that we have to take a piece of a bread and moisten it with water, nothing but we have to sprinkle some water in it. So on that surface of that bread mold, leave it in a warm place for 2 to 3 days till fluffy patches appear on them. Observe the patches under magnifying glass or microscope. So microscope is available in that is the biology lab. So you can see it, we can do it and we can see it. So or you can use also, you can use a magnifying glass, we will be will available in the physics lab, we can see it. Okay, cotton like threads are seen on the bread, cotton like a threads. Are you getting my point? Cotton you already know, in Kannada we can say that is a cotton, nothing but is a hatti, that is cotton like a structure. So these cotton like a structure will be appearing after 2 to 3 days on the bread mold, bread piece, that is nothing but a fluffy patches, fluffy patches. The cotton like threads are fungus bread mold, it is, these are cotton like a threads are nothing but these are fungus only. The bread mold is a saprotrope, that is we discussed what are saprotropes. Saprotropes does not contain chlorophyll, these are heterotropic mode of nutrition, does not containing chlorophyll, will depending on other organisms for its food, like that here, which is saprophytic, that is a bread mold is a saprotropic, that is saprophytic also we can say, saprotropic also. Okay, so bread mold initially does not have any chlorophyll to prepare its own food, which will be depending on other organisms. Then which is other organism? Fungus is the other organism. Are you getting my point? So fungus which will be appear on the bread mold, and bread mold and the fungus here, they are acting as a saprotropic relationship. They are playing a saprotropic relationship. That is, saprotropes get their food from dead and decaying organic matter. The saprotrope here acting as a bread mold, and they will get their food on growing on the dead and decaying organic matter. Their food they are getting it from dead and decaying organic matter. Also good example for this one is, that is which one? We discussed this saprotropes. I will go back to the previous slides. See this saprotropes. What do you call this one? You can just recall this. What do you call this one? This one. These all. Where you saw this? Now rainy season is started. You can see nearby your house any wooden stick or something, that is a big wooden piece, if it is kept nearby your house or besides of your house, wherever it is, whenever you move to the school, you can see so many, that is mushrooms will grown on the upper surfaces or its near surfaces, means they will grow on the dead and decaying matter. These mushrooms are acting as saprotropes. Initially does not containing chlorophyll, will not prepare their own food, will depending on the other organisms like that is dead and decaying matter. So they will get their food from dead and decaying matter by absorbing its food from those dead and decaying matter. In the same way here also what we discussed, that is the activity 1.2, that is also it is explaining saprotropic relationship between bread mold and this fungus. The fungus where they are getting food, that is bread mold is getting its food from the fungus, that is funguses are growing on the it's a dead and decaying matter, then the bread mold is getting its food. So this is a relationship between the bread mold and the fungus. In this way it will work. So this is a saprotropic mode of nutrition will comes under in a heterotropic mode of nutrition. So here we completed the one lesson, completely one lesson. If there is any questions, any queries, you can please drop that question into my personal number, not in the group. You can drop me that question in my personal number and I will come back to you with the solutions. And after this one I will give homework also. Thank you so much. That I don't want to suggest to again and again, that is you can just share this video in a Facebook, Twitter and other modes of, that is in the medias, so which will help me lot. That is I can lastly say that is subscribe to my channel RSK. You can go to the YouTube, you can search RSK competitive word. All my that is videos are available in my YouTube channel. You can go and you can subscribe, you can share to your friends, you can tell them, then inform them which are there studying in the 8th standard, that is 7th standard, and also it will be helpful for the 6th standard also. You can share to your friends which are learning in other CBSE schools, ICSE schools and state schools, all the you can share it. Okay, thank you so much.